Hey guys, welcome back to another episode of my channel. My name is Keika. I'm going over Campbell's biology textbook, and in this particular video, we'll be going over concept 5.2 of chapter 5. Carbohydrates serve as fuel and building material. So, carbohydrates include sugars and polymers of sugars. The simplest carbohydrates are the monosaccharides, or simple sugars. These are the monomers from which more complex carbohydrates are built. Disaccharides are double sugars, consisting of two monosaccharides joined by a covalent bond. Carbohydrate macromolecules are polymers called polysaccharides, composed of many sugar building blocks. So for my notes, I wrote carbohydrates include sugars and polymers of sugars. A monosaccharide is a simple sugar or a monomer, which is, again, right, a monomer is a building block. It's one part that chains together with other things. So as we'll see with a disaccharide, is two monosaccharides joined together by a covalent bond. So it's two monomers, which are the monosaccharide. And then a polysaccharide is a macromolecule polymer built up of multiple monosaccharides, more than two. So if it were two, it'd be a disaccharide. If it were more than two, it's a polysaccharide. Sugars. Monosaccharides, from the Greek monos, meaning single, and saccharine, meaning sugar, generally have molecular formulas that are some multiple of the unit CH2O. Glucose, the most common monosaccharide, is of central importance in the chemistry of life. In the structure of glucose, we can see the trademarks of a monosaccharide. The molecule has a carbonyl group and multiple hydroxyl groups. Depending on the location of the carbonyl group, a monosaccharide is either an aldose or a ketose. Glucose, for example, is an aldose, and fructose, an isomer of glucose, is a ketose. Another criterion for classifying monosaccharides is the size of the carbon skeleton, which ranges from 3 to 7 carbons long. Glucose, fructose, and other sugars that have 6 carbons are hexoses. Trioses are 3 carbon sugars, and pentoses, 5 carbon sugars, are also common. Okay. Still, another source of diversity for simple sugars is in the way their parts are arranged spatially around asymmetric carbons. Recall that an asymmetric carbon is a carbon attached to 4 different atoms or groups of atoms. Glucose and galactose, for example, differ only in the placement of parts around one asymmetric carbon. What seems like a small difference is significant enough to give the two sugars distinctive shapes and binding activities, thus different behaviors. Although it is convenient to draw glucose with a linear carbon skeleton, this representation is not completely accurate. In aqueous solutions, glucose molecules, as well as other 5 and 6 carbon sugars, form rings, because they are the most stable form of these sugars under physiological conditions. Monosaccharides, particularly glucose, are nutrients for cells. In the process known as cellular respiration, cells extract energy from glucose molecules by breaking them down in a series of reactions. Not only are monosaccharides of major fuel for cellular work, but their carbon skeletons also serve as raw material for the synthesis of other small organic molecules, such as amino acids and fatty acids. Monosaccharides that are not immediately used in these ways are generally incorporated as monomers into disaccharides or polysaccharides. Discuss next. A disaccharide consists of 2 monosaccharides joined by a glycosidic linkage, a covalent bond formed between 2 monosaccharides by a dehydration reaction. Glyco refers to carbohydrate. For example, maltose is a disaccharide formed by the linking of two molecules of glucose. Also known as malt sugar, maltose is an ingredient used in brewing beer. The most prevalent disaccharide, however, is sucrose, or table sugar. Its two monomers are glucose and fructose. Plants generally transport carbohydrates from leaves to roots and other non-photosynthetic organs in the form of sucrose. Lactose, the sugar present in milk, is another disaccharide. In this case, a glucose molecule is joined to a galactose molecule. Disaccharides must be broken down into monosaccharides to be used for energy by organisms. Lactose intolerance is a common condition in humans who lack lactase, the enzyme that breaks down lactose. The sugar is instead broken down by intestinal bacteria, causing the formation of gas and subsequent cramping. The problem may be avoided by taking the enzyme lactase when eating or drinking dairy products, or consuming dairy products that have already been treated with lactase to break down the lactose. Okay, so for sugars, I wrote monosaccharides have molecular formulas that are some multiple unit of CH2O. Glucose is the most common monosaccharide, consists of carbonyl groups and multiple hydroxyl groups. Depending on the location of the carbonyl group, a monosaccharide can be an aldose or ketose. Classification of monosaccharides also depends on the size of the carbon skeleton, which can be 3 to 7 carbons long. And common types of sugars are hexoses, trioxes, and pentoses, which depend on the number of carbons that are within the skeleton. Another source of diversity for simple sugars is in the way their parts are arranged spatially around the asymmetric carbons. And an asymmetric carbon is a carbon attached to 4 different atoms or groups of atoms. In aqueous solutions, glucose and other simple sugars form rings because they are the most stable form of these sugars in these conditions. Monosaccharides, like glucose, are major nutrients for cells and serve as raw material for synthesis of other types of small organic molecules, like amino acids and fatty acids. If not for fuel or synthesis of small organic molecules, they are also monomers for disaccharides or polysaccharides. Cellular respiration is when cells extract energy from glucose molecules by breaking them down in a series of reactions. And glycosidic linkage is a covalent bond that forms between 2 monosaccharides by a dehydration reaction. Examples of disaccharides include maltose, which is 2 glucoses together, sucrose, which is glucose and fructose, and lactose, which is glucose and galactose. Lactose intolerance is when humans lack the enzyme lactase, which is meant to break down lactose. Lactose gets broken down by intestinal bacteria instead, leading to the formation of gas and cramping. The solution is to take the enzyme lactase or consume dairy products that have been treated with lactase. Okay? Polysaccharides are macromolecules polysaccharides are macromolecules. Polymers with a few hundred to a few thousand monosaccharides joined by glycosidic linkages, which, as we mentioned before, is a covalent bond that forms between 2 monosaccharides by a dehydration reaction. And some polysaccharides serve as storage material, hydrolyzed as needed to provide monosaccharides for cells, and other polysaccharides serve as building material for structures that protect the cell or the whole organism. The architecture and function of a polysaccharide are determined by its monosaccharides and by the position of its glycosidic linkages. So, for polysaccharides, I wrote, polysaccharides are macromolecules, polymers with few hundred to a few thousand monosaccharides joined by glycosidic linkages, serves as storage material to be hydrolyzed as needed to provide monosaccharides per cell. and I'm just going to write energy. And it serves as a building material for structures that protect the cell or whole organism. Architecture and function of polysaccharides depend on monosaccharides and position of its glycosidic linkages. Storage polysaccharides. Both plants and animals store sugars for later use in the form of storage polysaccharides. Plants store starch, a polymer of glucose monomers, as granules within cellular structures known as plastids. Plastids include chloroplasts. Synthesizing starch enables the plant to stockpile surplus glucose. Because glucose is a major cellular fuel, starch represents stored energy. The sugar can later be withdrawn by the plant from this carbohydrate bank by hydrolysis, which breaks the bonds between the glucose monomers. Most animals, including humans, also have enzymes that can hydrolyze plant starch, making glucose available as a nutrient for cells. Potato tubers and grains, the fruits of wheat, maize, or corn, rice, and other grasses are the major sources of starch in the human diet. Most of the glucose monomers in starch are joined by 1 to 4 linkages, number 1 carbon to number 4 carbon, like the glucose units in maltose. The simplest form of starch, amylose, is unbranched. Amylopectin, a more complex starch, is a branch polymer with 1 to 6 linkages at the branch points. Both of these starches are shown in figure 5.6. We're just going to take a quick look. So we see A, starch. Storage structures, plastids containing starch granules in a potato tuber cell. So this is it, and this is what it looks like. So we have amylose, which looks like this. It has the glucose monomers. And we have amylopectin, which is somewhat branched. I'm not sure if this is amylopectin. Or, I'm not sure. Okay, let's go back. Okay. Animals store a polysaccharide called glycogen, a polymer of glucose that is like amylopectin, but more extensively branched. Vertebrates store glycogen, mainly in liver and muscle cells. Breakdown of glycogen in these cells releases glucose when the demand for energy increases. The extensively branched structure of glycogen fits its function. More free ends are available for breakdown. This stored fuel cannot sustain an animal for long, however. In humans, for example, glycogen stores are depleted in about a day, unless they are replenished by eating. This is an issue of concern in low-carbohydrate diets, which can result in weakness and fatigue. So we're going to take a look at glycogen in this figure. So as you can see, glycogen granules stored in muscle tissue. And as you can see here, the starch is a little simpler looking, right? It has 2 ends here and a little, for amylopectin, it has a branch. It's somewhat branched. But for glycogen, it's like it has 1, 2, 3, 4, 5. It just has more branches. And this is cellulose. We'll get to that later. Okay, so for storage polysaccharides, I wrote plants and animals store sugars for later in the form of storage polysaccharides. Plants store starch, a polymer of glucose molecules, as granules within cellular structures called plastids. Glucose is a major cellular fuel. Starch equals stored energy. Plants can withdraw sugar through hydrolysis, which breaks bonds between glucose monomers. Animals, including humans, have enzymes that hydrolyze plant starch. Sources of starch include potato tubers and grains, the fruit of wheat, corn, rice, and grasses. Some starches have 1 to 4 linkages and can either be branched or unbranched. Animals store glycogen, a polymer of glucose, which is mainly found in the liver and muscle cells, and the breakdown of glycogen into glucose occurs when there's a demand for energy. This, however, cannot sustain an animal for long. It lasts a day unless the animal eats. Low-carb diets result in weakness and fatigue. And structural polysaccharides. Organisms build strong materials from structural polysaccharides. For example, the polysaccharide cellulose is a major component of the tough walls that enclose plant cells. Globally, plants produce almost 10 to the 14th kilograms, or 100 billion tons, of cellulose per year. It is the most abundant organic compound on Earth. So going back to this figure, as we can see here, this is cellulose, and it looks like this. Cellulose microfibrils in the plant cell wall. A microfibril is a bundle of about 80 cellulose molecules. So as you can see, it's unbranched, it's pretty linear. Cellulose molecule unbranched, and hydrogen bonds between parallel cellulose molecules hold them together. Okay? So these are the different polysaccharides. Like starch, cellulose is a polymer of glucose with 1 to 4 glycosidic linkages, but the linkages in these two polymers differ. The difference is based on the fact that there are actually 2 slightly different ring structures for glucose. When glucose forms a ring, the hydroxyl group attached to the number 1 carbon is positioned either below or above the plane of the ring. These two ring forms for glucose are called alpha and beta respectively. In starch, all the glucose monomers are in the alpha configuration, the arrangement that we saw in figures 5.4 and 5.5. In contrast, the glucose monomers of cellulose are all in the beta configuration, making every glucose monomer upside down with respect to its neighbors. The differing glycosidic linkages in starch and cellulose give the two molecules distinct three-dimensional shapes. Certain starch molecules are largely helical, fitting their function of efficiently storing glucose units. Conversely, the cellulose molecule is straight. Cellulose is never branched, and some hydroxyl groups on its glucose monomers are free to hydrogen bond with the hydroxyls of other cellulose molecules lying parallel to it. In plant cell walls, parallel cellulose molecules held together in this way are grouped into units called microfibrils. These cable-like microfibrils are a strong building material for plants and an important substance for humans because cellulose is the major constituent of paper and the only component of cotton. The unbranched structure of cellulose thus fits its function, imparting strength to parts of the plant. Enzymes that digest starch by hydrolyzing its alpha linkages are unable to hydrolyze the beta linkages of cellulose due to the different shapes of these two molecules. In fact, few organisms possess enzymes that can digest cellulose. Almost all animals, including humans, do not. The cellulose in our food passes through the digestive tract and is eliminated with the feces. Along the way, the cellulose abrades the wall of the digestive tract and stimulates the lining to secrete mucus, which aids in the smooth passage of food through the tract. Thus, although cellulose is not a nutrient for humans, it is an important part of a healthy diet. Most fruits, vegetables, and whole grains are rich in cellulose. On food packages, insoluble fiber refers mainly to cellulose. Some microorganisms can digest cellulose, breaking it down into glucose monomers. A cow harbors cellulose-digesting prokaryotes and protists in its gut. These microbes hydrolyze the cellulose of hay and grass and convert the glucose to other compounds that nourish the cow. Similarly, a termite, which is unable to digest cellulose by itself, has prokaryotes or protists living in its gut that can make a meal of wood. Some fungi can also digest cellulose in soil and elsewhere, thereby helping recycle chemical elements within Earth's ecosystem. Another important structural polysaccharide is chitin. Chitin? The carbohydrate used by arthropods, insects, spiders, crustaceans, and related animals, to build their exoskeletons, hard cases that surround the soft parts of an animal. Made up of chitin, embedded in the layer of proteins, the case is leathery and flexible at first, but becomes hardened when the proteins are chemically linked to each other, as in insects, and encrusted with calcium carbonate, as in crabs. Chitin is also found in fungi, which use this polysaccharide rather than cellulose as the building material for their cell wall. Interesting. Chitin is similar to cellulose with beta linkages, except that the glucose monomer of chitin has a nitrogen-containing attachment. Okay. Alright. So, structural polysaccharides. Cellulose is a major component of the tough walls that enclose plant cells, and is the most abundant organic compound on Earth. Linkages in cellulose differ from starch. Still has 1 to 4 linkages, but has different ring structures for glucose. The hydroxyl group attached to the number 1 carbon is positioned either above or below the plane of the ring, resulting in either an alpha configuration or a beta configuration. Starch is in alpha configuration, while cellulose is in beta configuration, which is upside down. The difference of glycosidic linkages in starch and cellulose give them 2 molecules distinct 3-dimensional shapes. Certain starches are largely helical, while cellulose molecule is straight and never branched. The unbranched structure of cellulose fixes its function to add strength to parts of the plant. Enzymes that digest starch by hydrolyzing its alpha linkages cannot hydrolyze beta linkages in cellulose. Few organisms possess enzymes that required to digest cellulose. Almost all animals cannot digest cellulose. It passes through the digestive tract and it gets excreted as feces. Cellulose is not a nutrient, but is important for a healthy diet. Fruits, vegetables, whole grains, and is noted as insoluble fiber on nutritional labels. Some microorganisms can digest cellulose, breaking it down to glucose monomers, such as cows that have cellulose-digesting prokaryotes and protists in its gut, so it can hydrolyze the cellulose of hay and grass. Termites have prokaryotes or protists living in its gut that can digest wood, and some fungi can digest cellulose in soil. Another structural polysaccharide is chitin. Chitin. I think it's chitin. Yeah, I've been saying it wrong, I think. Chitin. Used to build exoskeletons by insects, spiders, crustaceans, etc. Also found in fungi for cell walls. It's similar to cellulose with beta linkages, but has a nitrogen-containing attachment. Okay. So, that's it for concept 5.2. Carbohydrates serve as fuel and building material. We went over sugars, polysaccharides, and the different types. So, next video we'll be going over concept 5.3, Lipids are a diverse group of hydrophobic molecules. Okay, so I hope you guys understood the material and that I helped you guys understand. Good luck with your studies. Please like and subscribe if you like, and I'll see you guys next time. Bye.